This is the possibilities of genetic engineering. The genetic engineering basis. In recent history, scientists have discovered and learned a new tool called genetic engineering. Genetic engineering allows you to cut variations of DNA and replace them with others. Genetic engineering has the power to affect all organisms. The most common organisms that will be affected by gene editing will be plants, bacteria, humans, and even animals. With gene editing rising into our current society, it has the power to play a massive role in the future. As of now, more research still needs to be done to ensure the safety of this procedure. Doctors are developing this technology to hopefully one day use it as a great and positive tool for our future. On the next slide. CRISPR. Now I'll introduce the term CRISPR. CRISPR is a technology that has been created as a new way to introduce gene editing. CRISPR, as you can see, stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR can be found in bacteria most often. This new tool will hopefully allow scientists to alter the DNA of those organisms. CRISPR will find a specific piece of DNA in the cell, cut that portion of the DNA, and insert a piece of DNA that it wants. CRISPR has been famously used by a doctor named He Jiankui. This doctor used it on twin girls. When CRISPR is conducted, every cell is affected, including reproductive cells. Society today still doesn't know the long-term effects of CRISPR. The possibilities of CRISPR can be endless, but will it be used in a positive or negative way? On the next slide. We will now be talking about the main branches of gene editing. And the first branch will be somatic gene editing. Somatic gene editing has to deal with only the effects of the patient being treated and some of his or her cells. Somatic gene editing involves the modification of the patient's DNA in order to treat what is necessary. That has been caused by genetic mutation. In somatic gene editing, the reproductive cells are not touched. Somatic gene editing is less powerful than the other type of gene editing. Somatic gene editing is, as of now, known as the safest type of gene editing. On to the next main branch. The next main branch is called germline gene editing. Germline gene editing affects every single cell within the organism being operated on, including the egg and sperm cells, which can be passed on to others. A perfect example of germline gene editing is Jiankui's procedure on those two twin girls. Germline gene editing takes its place in altering the genome as a human embryo at the earliest possible stage. Germline gene editing is the most powerful editing tool out of germline and somatic editing. Once the germline process has been done on an organism, the organism has the power to pass on its edited cells to its children or babies. With so much power being held in genetic engineering, unknown risks play a massive role in its future. As of today, germline editing is banned in the United States. Many wonder if the advantages of germline editing will outweigh the disadvantages. On to the next slide. The social and ethical questions on gene editing. In this slide, I will discuss the new discovery and how it comes up with new questions. These questions are very delicate because of what they are dealing with. These questions are so hard to answer because it deals with more than just an individual. Many people feel as though gene editing will never be proven safe to use. The outcomes and possibilities could harm and negatively affect our future as a society. New forms of discrimination, equality, and even societal classes may be created. With humans having the ability to change who they are, this can create an imbalance in society. Because of all these negative possibilities, 40 countries have banned germline editing. Now, what does this mean for Earth society as a whole? What type of final decisions will be made? For now, we don't know, but the future of gene editing may not end up as some imagine it will. On to the final slide. Finally, who will have the final say on this matter? DNA editing will have an effect on how, as a society, we want our future to be. Gene editing has a significance not only on individual humans, but has extreme potential on society. The decision on gene editing cannot be made by a small group or corporation. When it comes down to a decision as large as this, it must be a worldwide decision. As a global society, we should all be in this together. Globally, we must act on new discoveries with diligence. This will help ourselves figure out the positive and negative possibilities of the discoveries, as well as the impact to society. Gene editing and DNA modification is an extreme issue and must be acted on accordingly.